Welcome all. So, please allow me to take honor in introducing two distinguished speakers, Dr. Amita Sharma and Harshita Bhargava, who are assistant professors, senior grade at IIS University, Jaipur. They have at least 30 years of vast experience in between them, working on various facets of machine learning, artificial intelligence, cloud-based infrastructure, and as well as the recommender-based systems, which Harshita especially has expertise in. Amita and Harshita, they have been working together on various aspects of feature selection-based algorithms as well, extracting computational capabilities towards machine learning and as well as artificial intelligence based. They've also come up with a kind of a novel case study based approaches. So today they are really going to take us to the next generation models, taking the recommendation systems for biology and medicine. So please feel free to ask some questions. They have been experts in their own fields and they have been kind enough to have spent time last time as well during the previous lockdown time. And the floor is yours, Harshita and Amita. Thank you so much.
Thank you so much, sir. I request Harshita ma'am to just share the presentation so that we can start.
Sharma.
I hope slides are visible to you. Okay, now let's start with the topic, recommender systems. Basically, as we move in this particular topic, I just give you the brief introduction about the content we're going to cover in this workshop. We have decided four things which we're going to focus upon. Firstly, data science and biology, how we are actually moved from data science and taking the biology as a case study, and then we try to develop different systems and how recommender system happened to us. As well as we'll take some practical example of Python and a very interesting tool which is Orange tool, which could be a beneficial for all the beginners who want to start working in machine learning or recommender system. Um, Next slide. Yes. So basically, as we all know, we in case of data science and biology, we have data, huge set of data, and also we have certain kind of algorithm, which is basically computational algorithms, work upon data, and also we have certain kind of mathematical and statistical modeling equations and algorithms which can be used for various types of analysis, and with those kind of analysis we generate different kind of knowledge. So all these work together and we form a computational biology, and the core essence of this particular application or particular area is analysis. What we analyze in our data, we transform into a knowledge, and that knowledge is further being used in different kind of application to solve different kinds of problems, and as also to find the better solution of existing problem. Next slide. So if we talk about the type of analysis, in general, we have four kinds of analysis. Descriptive analysis, which generally focuses on what happened. And then we have diagnostic analytics, which works upon why did it happen. And then we have predictive analytics, which is quite popular these days, and where we usually apply all kind of machine learning algorithms, which generally gives us what will happen, which helps in forecasting our future, and as well as finding the certain kind of solutions to our existing problems, and with that prediction we can find a way out or pathway for our problem. Then the fourth, which is very important, is prescriptive analytics, which help us to plan our actions so that we can get the better results. Till predictive analytics, it is just general information about the happening or the event, but in prescriptive analytics, we give the suggestions or a prescription so that we can improve the event by giving the better inputs or by creating the environment in such a way that we get the better results. So basically, prescriptive analytics focus on how can we make it happen? How can we improve our environment or event by focusing on a particular set of actions? So the whole recommender system is based on prescriptive analytics. Next slide. As we all know, in predictive analysis, there are two kind of stages. First stage, we have historic data. We're going to pre-process that data and then we apply some predictive algorithms and form a model. Okay, it is very simple and we usually do all these kind of things with machine learning algorithms. In second stage, what we do, we take some test data just to prove it or to check whether the model is working accurately or efficiently or not. We that then provide that particular set of data to our model and then we get different kinds of predictions. So it is very simple example of how we perform a predictive analysis. Now in case of prescriptive analysis, next slide, we have several kind of data. Like we have data which we derive from the descriptive analysis. We have data which we gathered from diagnostic analytics. Also, we have data from prediction. So all these kind of data all are taken together and then based on that data, based on the observation gathered from that data, we generally try to find the better solutions or find the actions which can lead to a good or far more better solution for our problem. Okay, so that we can plan the action accordingly by observing all the data. Next slide. So how prescriptive analytics works? We have different kinds of observed data. We gather it either from descriptive analysis or we have raw kind of data also. Sometimes we have some data from predictions. We have data from diagnostic. All kind of data is taken care in this analytics. And we merge those data or we can combine those data based on the similarity or association relationship. Okay. In similarity, we find the similar pattern or in association case, we find the relationship in between their features or their behavior present in a particular case of data. Based on that similarity and an association, we create a model. That model generates certain kind of recommendations or certain kinds of rules. On based on those rules, we can plan our action. So basically, in this model, what we are doing, we are gathering data from various channels. We are putting it into our database or in data warehouse or in a cloud, and then we apply a similarity algorithm, association algorithms, or there are number of algorithms which we'll take further in the same presentation. What kind of algorithm is basically used for prescriptive analysis? Those algorithms are applied and model is created, and by understanding the what kind of data we have, model focuses on understanding of the data. So after that, we just generate the recommendations. Recommendations on the base of recommendations, we plan the action. This action is being implemented, and from that implementation, we again generate certain kinds of feedback so that we can check whether the actions which we have planned is appropriate or not, whether it is improving our result or not. So after doing that, with this feedback, we again try to remodel the or restructure the uh, particular problem and get further more recommendations. So basically, the recommendation or prescriptive analytics work in a looping format. We have to constantly validate the recommendations which we are putting in our case study. So this is basic idea how a prescriptive analytics and the recommendation systems are being initiated and how it is useful for bringing out the good solution for our case study or for our problems. Next slide. So now I request Harshita ma'am to just take over the session and explain further what kind of algorithms are there in recommender system and how it is different from expert system, because we already have the expert system and still we are running with the recommender system. What is the minute or major difference between the recommender system and expert system? It should be taken care before starting working in this particular area. Okay, Harshita, over to you.
Okay. Thank you, Amita ma'am. Am I audible there?
Yes, you are audible.
Okay. Okay. Basically, when we talk about the recommender systems, everybody must have used it knowingly or unknowingly. You must have read about these sentences like customers who bought this item also bought. You have read the sentence in Amazon basically, and people like you who bought pen drive also bought the USB cable. So this is basically the association rule mining that they have applied in their recommendation algorithm. So when we talk about the recommendation systems, initially you can say when we talk about the internet, it provides you a variety of choices in the form of research articles, in the form of some information. It can be movies, it can be audios, it can be videos, it can be music, etc., etc. So these are all the items or the services that are provided on the internet. Now, what was the initial pattern was you browse for these items through the websites or through the Google search engines, like search engines we हम use करते हैं. For that purpose, we filter out the items and that are suitable for our as per our choices, as per our parameters. We select the particular product or we select the particular item. It can be a movie, it can be music also, but that took time. Okay, for this purpose, recommendation systems were employed. Now, what they do is they perform automatic filtering. They take up your feedbacks, and that feedback may be in the form of ratings. Suppose you have purchased an item and you have provided a star rating to that item. Now, this kind of feedback will be utilized further for generating the recommendations for similar kind of products. So, basically, what it does, it depends upon the domain wherein you are applying that recommendation system. So, it depends what kind of algorithm you are using, but what happened was this automatic filtering feature was introduced that obviously they came to the rescue, and it provided you a set of search options, set of relevant choices that were far much lesser than the whole domain. Okay, so you have an enormous amount of information. उसमें से हमको एक filtered output मिला है. That filtered output can be used and it can be used for making decisions. So basically, what we think is many choices may lead to wrong decisions, but if we employ a recommender system inside any of the domain, it may lead to good decisions. Okay, so for that purpose, we use such kind of systems. Now, if we talk about a system is basically a big word. Of course, we can't we can't give a demo on implementing a recommender system, of course, in one 2 hour workshop. But of course, if we talk about a system is basically a set of components. Okay, these components work in synchronization and they work for a common goal, of course. So हम अगर recommendation systems की बात करते हैं तो basically what they do is they provide the set of related relevant recommendations to the end users. So if we talk about what are the entities that are involved, so if I relate it to the e-commerce domain, many of you must मतलब आप उससे थोड़ा सा relate कर पाएंगे. So if we talk about the e-commerce domain, we have two set of entities. One is the set of items, and the second is the set of users that are there in that domain for purchasing those kind of items, or they are providing some kind of feedback in the form of some ratings. So if I buy some product, I provide a simple relevant rating to that product. ठीक है, वो कुछ भी हो सकती है. इट कैन बी अ स्टार रेटिंग, इट कैन बी इन द फॉर्म ऑफ अ लाइक बटन, जैसे फेसबुक में होता है. इन द फेसबुक यू प्रोवाइड अ लाइक if you like that particular recommendation. तो वो यहाँ पर क्या है, basically that is a kind of feedback that is being provided to that system, and that feedback is used for the future recommendations. ठीक है, तो these predictions can be done appropriately by your feedbacks. Now this can be an explicit feedback, जैसे हमने कहा कि suppose you provide a rating, so that is an explicit feedback. Suppose you provide आप like button यहाँ पे press करते हैं, तो that like is basically a Facebook like is an intimation, it is an indication कि you like that recommendation. ठीक है, जो भी आपने यहाँ पे friends की list यहाँ पे दी है, तो you like that particular post or anything like that. So these are the kind of explicit feedbacks that this system uses. In along with these, we also have a set of implicit feedbacks. Now, what are implicit feedbacks? Like how much time you have spent on a website or viewing that particular kind of product. ठीक है, वो session based भी हो सकता है ना? It can be a contextual also, contextual, session based. These are the mixed, मतलब ये दोनों mixture हैं, but they provide a kind of implicit feedback. ठीक है, जो यहाँ पर हम दे सकते हैं, but that is an indirect feedback. It's not provided by the user itself. ठीक है, you purchase an item. This is also a kind of feedback that you have provided to the system. So these kind of feedbacks are aggregated and then the recommendations are generated. So this is the system that is deployed. But underlying a recommender system, the most important component that works is the recommendation algorithm that you use. Okay, so this can be, it can be a model based, it can be a machine learning based algorithm, it can be out of these four categories too. ओके इसके अलावा यहाँ पे कुछ कॉम्बिनेशंस भी हम एम्प्लॉय कर सकते हैं. वी कैन गो फॉर अ हाइब्रिड वन. ओके सो आई विल कम वन बाय वन to each of these techniques because ऐसे text आपको समझ में नहीं आएगा, but we'll start with a collaborative one because this is the most popularly employed or this collaborative filtering algorithm that has been employed in most of the websites in the e-commerce itself. If we talk about Amazon, in Amazon you can see these kind of text there frequently bought together. These two items are related, that's why they are frequently bought together. So they are trying to increase their sales. So collaborative filtering works on the wisdom of crowd basically. What other users have recommended, what other users have bought, ठीक है, उसके हिसाब से आपको recommendations दिए जाते हैं. तो these are the filtered recommendations and they are based on what others have liked. ठीक है, तो हम यहाँ पे मानते हैं similar users will have tend to have similar taste in the future too. Okay, or you can say similar kind of items. Basically, it employs the item to item based collaborative filtering. So we can say similar kind of items will have the similar kind of users that will rate. Now similar kind of items का मतलब क्या है? The similarly rated items. ठीक है, अगर हम कहते हैं कि यहाँ पे this book also has the similar kind of rating with the same user, ठीक है, इस पे second item also has the similar kind of rating. So these are the similar kind of users, ठीक है, जिसको हम यहाँ पर recommend कर सकते हैं. ठीक है, तो यहाँ पर हम क्या कह सकते हैं? Customers who bought this item also bought. So this is basically an item to item collaborative filtering. They find out in the underlying algorithm, what they do is they find out the similarities between the item for each item. ठीक है, हम यहाँ पर अगर मैट्रिक्स की बात करें तो हमारे पास item 1 से लेकर n तक यहाँ पर items है. Similarly on the column also you have item 1, 2, 3 and n तक यहाँ पर items होंगे and we have an item to item similarity that is being calculated. ठीक है, if we talk about Amazon, Amazon calculates, मतलब they can pre-calculate, हम यहाँ पे ये पहले से करके रख सकते हैं. This can be done offline. So this is an offline computation because you have several thousand millions of items there. तो आप यहाँ पर क्या कर सकते हो? You can calculate these similarities at the initial level in the offline manner. Okay, and these similarities often do not change. तो हम यहाँ पर मानते हैं कि items आपके कुछ हद तक fix रहते हैं, but the number of users can be changed. Number of users आपकी obviously बात है, they would be increasing. ठीक है, डे बाय डे. तो ये स्केलेबिलिटी एक इशू रहेगा. स्केलेबिलिटी कैसे है? बिकॉज यू कैन सी, यू कैन इमेजिन इफ यू हैव n number of users and m number of items, so it's very obvious each user will not be rating each and every item. Okay, and even if I am the user, I'll not be visiting each and every item in the future also. So there are you can say कि initial level पे हम बात करें तो we have a sparse kind of matrix that is available there, and there should be solutions, there should be ways how to overcome the sparsity issue. Okay, so this has to be sparse because every user will not be rating each and every item. So this is one of the issue that has to be taken care of while implementing such kind of algorithm. Okay, and the next one is the content-based filtering algorithm. If you have heard of Pandora Music, it is an online podcast. It has an app also. आप web version भी use कर सकते हैं इसका. You can have an online application also. तो आपके पास app, mobile app. ये basically क्या करता है? It basically creates a music app list. ठीक है, म्यूजिक का आपका स्टेशन प्ले स्टेशन क्रिएट करता है depending upon your choice of artist or the attributes or the type of songs that you want to listen. So this is a kind of personalized playstation that will be created by this kind of app. So ये कैसे create होता है? That depends upon the features that you have listed. Suppose जैसे यहाँ पे this is the initial level when you haven't checked in, you haven't created, you haven't logged in your in this app. तो यहाँ पर this is not available in India basically. It is a US based application, but in इसमें initial level पे what they say is you have to specify these kind of things. जैसे आपने यहाँ पे artist बताया और you have provided a song, so वो क्या करेंगे, they will create a play station, ठीक है, that is personalized, है ना. अभी personalized कैसे हुआ है? It is working on the features of that particular song. ठीक है, and they basically this Pandora Music, it's very popular because this is based on the Music Genome Project, है ना. तो यहाँ पर म्यूजिक के बहुत सारे फीचर्स हैं, दे हैव एग्रीगेटेड, दे हैव फाउंड आउट these are the set of features for each and every song. So what they do is they create an initial user profile, and after that with each and every song or each and every artist, they match these features, these content related features with your user profile, and they create a personalized or personalized playstation बनाते हैं. After that they keep on filtering that particular thing. So आपके पास क्या होगा, हमेशा म्यूजिक इज नॉट इक्वल टू ऑल ऑब्वियसली, है ना. तो दैट इज अ पर्सनलाइज वर्जन. तो पर्सनलाइज वर्जन आपको पंडोरा देता है, एंड इट वर्क्स ऑन द कंटेंट बेस्ड फिल्टरिंग टेक्निक that works on the content features of the items, not the ratings. ठीक है, तो this is one of the example, best example if you talk about the content based filtering. The next is the hybrid of the content based and the collaborative approach. In this, if we talk about the Netflix, now Netflix is basically using the combination of the content based and the collaborative approaches, in which you can see one line there, because you watched John Wick, that's why these recommendations have been provided. Now, this is a basic content-based recommendation because it is picking up what you had watched earlier, और उसमें से यहाँ पे they have picked up the artist, है ना. और इससे मैच करते हुए जितने भी मूवीज होंगी, these movies would be recommended. Of course, इसमें items क्या हैं? The item has been replaced by the movie. Now, movie is the item that is being recommended to the particular user. Okay, so it also provides you the collaborative approach. Collaborative में क्या होता है, जितने भी movies हैं, how similarly they have been ranked by the users, so that collaborative approach is also taken care of. So you always have a hybrid version working there, in which you utilize both these approaches, both the content based and the collaborative based. इसका और कोई example आप देखते हैं तो LinkedIn is also one of the best examples of the hybrid approach, in which they utilize both of these approaches, these algorithms, while recommending the items or the job profiles to you. The next one is the knowledge-based filtering. In the knowledge-based filtering, what happens is basically we don't have the ratings. Initial level ratings आपकी नहीं होती है. हम कह सकते हैं कि we particularly use such kind of filtering algorithms for those kind of items that are not frequently bought, or they are very expensive to be bought every now and then. For those purposes, we normally use the knowledge-based filtering. This basically, what it eliminates is, initially what it takes is, in the form of inputs, it takes some critics, or you can say it takes some inputs from the user. And on the basis of those inputs, they specify a particular recommendation that basically matches with your choice. Basically, जैसे यहाँ पर आपको इंटरफेस में दिख रहा होगा, this is a housing, it is a hypothetical example, that is a case based recommendation. It is for buying a home. And you specified the following target. Suppose you specified that I want a home at this particular location. Now, these are the recommendations that have been generated matching with these specific target, but with the specified list of choices जो आपने यहाँ पे दिया है कि I want it should be a smaller price, ठीक है, प्राइस कम होना चाहिए, द नंबर ऑफ बेडरूम्स मस्ट बी लाइक दिस, ठीक है, तो आप ये इंक्रीज डिक्रीज कर सकते हो, यू कैन डिफर द स्टाइल, हाउ द लोकेलिटी शुड बी, तो दीज आर द चेंजेस दैट यू कैन गिव एवरी नाउ एंड देन। सो जैसे जैसे आप यहाँ पे these critics or this information has been provided, automatically the recommendation would be updated. So this is a kind of knowledge based filtering. इसमें हम normally we employ a case base or a knowledge base in which we that can be also done in the form of an ontology. We talk about medical science या biology में अभी हम अगर बात करते हैं तो we can employ an ontology that is already been built, या आप अपनी खुद की भी ontology create कर सकते हैं. तो this is basically a knowledge based filtering in which it is either case based or it is constraint based. Okay. Next, if we talk about the recommendation system applications, we can say these are the popular domains wherein they have been applied. We have already talked about the e-commerce domain. In the e-learning, you can say it is technology enhanced learning. This is technically said like this. Technology enhanced learning means जैसे MOOC courses है या online you have several courses that are running at Coursera. They employ such kind of application system, recommendation systems in which depending upon your specialization, suppose you specify your area. I am from data science, I want to learn computer science or computer software engineering, like these kind of topics can be initially provided. Now these specializations are used to create your profile. Okay, and then after that, they can provide you the recommendations as soon as a new course is there on the Coursera. They will provide you the relevant recommendations. You can go for such a kind of course. So they are basically used in e-learning also. In the entertainment industry, Netflix is the best one of the best example that you can relate to. In the travel and tourism, you can see Trivago and such kind of websites are also there which employ such kind of recommendation system. Of course, in the travel and tourism, if you talk about this domain, you need to be very context specific. Okay, इसमें context awareness is very much needed in the recommendation system, because at what point of time you are going to travel, okay, which season is there, है ना, तो ये सारे contextual factors have to be kept in mind. So this is basically employed. These kind of things need to be done while implementing a recommendation system. It has to be context aware. So in the social networks, Facebook, okay, you have come across Facebook, Twitter, Instagram, okay, Pinterest. These are the basic social networks that employ such kind of recommendation systems. Of course, the technology that is being used, they may be employing some graph databases and all, because in the social networks you have a great amount of users that are connected together, and you should be able to recommend the users and every now and then, and they should be updated frequently, of course across the networks. The next is the web search. In the Google search also you found, you can see these applications. In the gastronomy based applications, you can see the health based RS, or you can see what kind of food should be taken, what kind of nutrient should be recommended to a particular user. So that depends upon the user profile. Also, you can have a content based recommendation system that can be deployed in this area. So this needs to be explored, and they have been implemented earlier also. And a very large number of deep learning applications, deep learning algorithms have been deployed for such kind of domains. Next is the drug discovery, precision medicine, and these are all related somewhat to the medicine. And of course, drug discovery and drug repurposing की बात करें, the most important one, and that is the hottest topic in the current scenario, is the drug repurposing, in which we want to utilize a specific we want to utilize an existing drug for curing some other disease other than that for which it was intended to. Initially, suppose आपने कहा कि a particular drug is being used to cure malaria. So if that drug can be repurposed to cure some other disease, then that is the best thing, because you can decrease the amount of drug discovery time that takes up around 10 years to come up with a feasible drug. So you can come up with the drug repurposing that is very much needed, and that is being very much used in the COVID scenario, because it is very difficult to come up with a drug, okay, that can cure a disease, or that can target a particular protein. Next, we can see
Coming back to the topic, if we say recommendation systems that are employed in medicine and biology, I'll just explain you the core concepts. Suppose in medicine, if we talk about the traditional way of recommendations, because recommendations are everywhere, okay. Initially हम कहते हैं जैसे wisdom of the crowd है ना, जो सबने कहा वही हम follow करते हैं, तो that basically implies to medicine also. ठीक है, though this is to be done very it has to be uh, supported by many factual things, but of course this is the traditional recommendation system that was earlier used, जो अभी भी use आता है, but of course this can be replaced by an automated one. Now what happens in the traditional medicine is you get some inputs in the form of basic demographics like age, height, weight, gender, your name, medical history, diagnosis, procedures that have been already applied, reports, etc. These are the inputs that are given to the doctor. The doctor takes his time, he performs some analysis, they provide the recommendations. Now, recommendations can be in the form of some medicine या screening के form में हो सकते हैं. ठीक है, these recommendations they basically they again help in early diagnosis. ठीक है, treatment हो जाता है and the patient is cured. Now, this is the traditional recommendation जो पहले यहाँ पे चलता था, अभी भी चलता है of course. Now this can be replaced by these automated engines via recommendation systems, in which what you can say is the same kind of inputs are being entered to an EMR system, electronic medical record system. Now this system will use a recommendation system, okay, while aggregating the data from all the other millions of patients that share the same kind of profile. सो इफ आई एम अ डायबिटिक पेशेंट मे बी आपका यहाँ पर आपका डेटा जो लिया जाता है दैट मे बी एग्रीगेटेड with several other such kind of patients जो भी डायबिटिक हैं and they are sharing the same kind of profile, है ना. प्रोफाइल में हो सकता है डेमोग्राफिक्स वगैरह भी आप कंपेयर करें. there are many parameters to it, but of course you can have the model building, of course, that is a machine learning model, or you can have some similarity calculation logic. How will you compare these two patients, है ना, और millions of patients से आप similarity कैसे निकालेंगे, उसके लिए you have several other parameters. ठीक है, हम क्या similarity यहाँ पे metric काम में लेंगे, या what kind of model building would be done. ठीक है, that can be done easily, ठीक है, through the use of the recommendation system, and you have the generated recommendations in the form of मतलब electronically आप यहाँ पे बता सकते हैं on the screen, on the interface you have such recommendations and they can be further used. Of course हो सकता है ये 100% सही नहीं हो, of course, but these recommendations will be much better than the traditional ones. बिकॉज ट्रेडिशनल में हम क्या कर रहे हैं, वन डॉक्टर इज ओनली सर्चिंग, वो भी क्या कर रहे हैं, ऑब्वियसली दे आर ऑल्सो थिंकिंग इन द माइंड कि यहाँ पर कितने लोग होंगे जो यहाँ पर पहले से हमारे पास आए हैं. दीज आर द डायबिटिक पेशेंट्स दैट हैव बीन दैट हैव कम टू अस. ठीक है, और यहाँ पर हमने ये मेडिकेशंस दिए थे, तो मोर और लेस आप क्या करते हो डॉक्टर्स, व्हाट दे डू इज, ठीक है, वो उसके हिसाब से यहाँ पे दे प्रिस्क्राइब यू सम मेडिसिन और ऑल. बट इसमें क्या होगा, यहाँ पर जो रिकमेंडेशंस आएंगी दे हैव बीन दिस डिसीजन, जो रिकमेंडेशंस ये आए हैं दे आर डेटा असिस्टेड. ठीक है, तो डेटा असिस्टेड रिकमेंडेशंस ऑफ कोर्स दे आर मच वेरी मतलब वैल्यूएबल and very much effective than the traditional ones. So this is basically that is being used in the medicine area also. You can go for I can suggest you मतलब CURAI, CURAI. This is a recommendation system, medical advice recommendation system that has been implemented by Xavier American. He worked in Amazon. He's a founder of this kind of system, जिसमें they have been using all such kind of algorithms. जिसमें यहाँ पर they use the medical ontologies also, the ICD system, ICD-10 system of diseases also. तो यहाँ पे they have used several algorithms. मशीन लर्निंग में टेक्स्ट माइनिंग भी उन्हें यूज़ किया है, बिकॉज हम बेसिकली क्या कर सकते हैं, वी कैन एक्सट्रैक्ट द थिंग्स फ्रॉम द रिसर्च आर्टिकल्स ऑल्सो, बट you can't go only for that only to that particular text mining area, है ना. तो आपको क्या करना पड़ेगा, you have to aggregate from all these things and then you can have the relevant recommendations that can be provided as a medical advice to the patients. Okay, there are several other websites also like Patients Like Me and all, where you can share your data there. Okay. So in medicine, likewise, this is the basic logic जो यहाँ पे employ किया जाता है. How to implement this? Behind these, you have some recommendation algorithms or the ML-based algorithms that have been employed. If we talk about the applications of RS in biology, these are the basic applications. I am interested in the last one because that is my research topic. That is related. We'll be implementing a recommendation algorithm or a model, okay, to predict the drug target interactions. Now, अगर हम first three की बात की कर रहे हैं, तो the last one is the basis for all these three. ठीक है, drug repurposing, whether it be the drug side effect prediction or it be the drug discovery or ठीक है, तो यहाँ पे ये जो तीन हैं आपके, these all applications are related with the drug target interaction prediction problem. So when I talk about the drug repurposing means you are using an existing drug to cure some other disease other than that for which it was designed. ठीक है, नेक्स्ट इज अ ड्रग साइड इफेक्ट प्रिडिक्शन. साइड इफेक्ट प्रिडिक्शन मीन्स अगर आपके पास यहाँ पर सपोज हम कहते हैं कि यू आर जैसे हम बात करेंगे इफ I talk about in terms of the drug targets, drug proteins. ठीक है, I come to this example. आप इससे समझ जाओगे यहाँ पे through this diagram. If we talk about this problem statement, how I relate the recommendation systems to this problem. Basically, we have the drug target interaction prediction or association problem. Now, what are the drugs and what are the targets? ठीक है, अगर हम इसको मॉडल करते हैं तो drug target interaction, or in short, I can say it is DTI problem. This can be molded or it can be modeled as a recommender task in which the drugs are the users. ठीक है, यहाँ पे these are the targets मतलब items are the targets. Now target specifically what I am talking about, these are the proteins. We'll be using the proteins but they may be RNA, DNA. ठीक है, तो यहाँ पे targets कुछ और भी हो सकते हैं. But if we talk about this kind of drug target interaction problem, basically what we have is we have n number of drugs and we have n number of targets. Each drug is connected with a target. इसका मतलब क्या है, where there is an edge there is an interaction. And in case of non-edge क्या मतलब बात करेंगे, if we talk about this bipartite graph, you must have heard about the adjacency matrix. So adjacency matrix specifies कि how each drug is related to, मतलब अगर हम यहाँ पर m nodes और n यहाँ पे आइटम्स की बात कर रहे हैं, m यूजर्स और n आइटम्स की बात कर रहे हैं, तो वी हैव ऑफ कोर्स काइंड ऑफ रेटिंग मैट्रिक्स. ठीक है, हम ई कॉमर्स में बात करते हैं तो वहाँ पर वी हैव अ रेटिंग मैट्रिक्स, या हम उसको यूटिलिटी मैट्रिक्स भी बोलते हैं. वी कैन से इट एज अ प्रेफरेंस मैट्रिक्स ऑल्सो. बट इन केस ऑफ ड्रग्स एंड टारगेट्स अगर हम कहेंगे तो यहाँ पर वी हैव द नोन एसोसिएशंस और द नोन इंटरेक्शंस दैट आर रिप्रेजेंटेड बाय दीज एजेस. Okay, so if we have association matrix or an interaction drug target interaction matrix, we can depict, मतलब graphically हमने इसको graph के form में किया है, but internally if we talk it about like this, we have an association matrix. ठीक है, association matrix में क्या होगा, each drug is related to a target or it is not interacting with the target of course. ठीक है, तो या तो edge होगी या नहीं होगी. If there is no edge, that means यहाँ पे will not be putting a zero there. ठीक है, दो in many studies there are non-edge is categorized as zero, but it is not zero, it is not being verified. You can say where there is an edge between a drug and a target, this edge has been detected, it has been taken up from some database. ठीक है, online जो open source databases हैं वहाँ पर हमारे पास ये information होगी. So the known and the verified and the validated interactions are there in the databases, but the not, मतलब जो यहाँ पे नॉन एजेस हैं, these have not been verified, है ना, validated नहीं है, तो जो negative interactions हैं you don't know about them. ठीक है, तो हम यहाँ पर जैसे D1 है, it is related to T1 but it is not interacting with T2, but this question is unknown, ये आपको पता नहीं है, it has not been validated कि whether there is an interaction or not. ठीक है, तो these are the unknown interactions. It may be an interaction, it may be a non-interaction. Okay, so if we represent this matrix, again it will be a sparse matrix. अब इसमें यहाँ पर कंसेंसस कैसे बैठता है, हम यहाँ पर मॉडल क्यों कर सकते हैं इसको एज अ रिकमेंडेशन टास्क, बिकॉज द रिकमेंडेशन सिस्टम में भी हमारे पास क्या होता है, वी हैव सर्टेन यूजर्स, वी हैव सर्टेन आइटम्स एंड इट इज ऑल्सो बायपार्टाइट ग्राफ काइंड ऑफ थिंग, जिसमें हर यूजर किसी आइटम से यहाँ पर if it is being rated, if this user has rated a particular item, so there is an edge, and if it is not rated an item, ठीक है, तो वहाँ पे एक एज नहीं होगी, द एज वुड बी मिसिंग, ठीक है। तो सिमिलर कंसेंसस मतलब हम यहाँ पे भी देख सकते हैं, तो यू कैन सी दैट द रिकमेंडेशन एल्गोरिथम्स कैन वर्क इन दिस प्रॉब्लम ऑल्सो. So if we talk about the classical ones, maybe the classical ones can prove मतलब यहाँ पर काम आ सकते हैं, but of course they have to be मतलब वहाँ पे उसको reframe करने की ज़रूरत है. We need to reframe those kind of algorithms. Yeah, you can see you can have the hybrid approach being applied to this kind of prediction problem. If we target मतलब हम अगर DTI की बात करते हैं तो DTI में क्या होगा? Basically, it can be framed as a recommendation task. ओके, तो हम ये कह सकते हैं वन मतलब प्रिंसिपल दैट कैन बी टेकन फ्रॉम द कोलैबोरेटिव फिल्टरिंग एल्गोरिथम कैन बी लाइक ड्रग सिमिलर इन स्ट्रक्चर आर लाइकली टू हैव सिमिलर टारगेट्स, ठीक है। इसी तरह से हम वाइस वर्सा भी कह सकते हैं द टारगेट्स आर सिमिलर इन सीक्वेंस लीड टू मतलब दे टेंड टू आर दे आर लाइकली टू हैव an interaction with the similar kind of drugs. So that can be a consensus. मतलब एक principle हम hypothesis लेके चल सकते हैं, but this hypothesis is not always true. So you need to have a model. Either it is a machine learning model or it is a hybrid kind of thing जो आप यहाँ पे implement करते हैं. So for that purpose हम यहाँ पे DTI problem दे सकते हैं and it can be modeled as a recommendation task. द नेक्स्ट थिंग इज हम यहाँ पे बात कर रहे हैं these are the basic मतलब प्रिंसिपल, अगर हम यहाँ पे प्रिंसिपली कहें तो DTI prediction problem forms the basis for polypharmacology in which you can see a drug multiple drugs act on multiple targets, ठीक है, एंड सिमिलरली वाइस वर्सा भी है, तो देयर इज अ मेनी टू मेनी रिलेशनशिप. जो पहले आपके पास वन ड्रग वन टारगेट वन डिजीज का मैकेनिज्म था, दैट पैराडाइम हैज बीन चेंज टू मल्टीपल ड्रग्स मल्टीपल डिजीज मल्टीपल टारगेट्स एंड मल्टीपल डिजीजेस, ठीक है. तो यहाँ पे देयर इज अ मेनी टू मेनी रिलेशनशिप that can be seen within the drugs and the targets. The next is the drug discovery, drug repositioning. Repositioning या drug repurposing हम कह सकते हैं. Predicting the side effect of the drugs. Side effects हम कैसे कहेंगे? Because if we say कि for a particular drug, the drug in question, we recommend a particular set of targets. ठीक है, some are knowns, ठीक है, हमने यहाँ पे जो लिस्ट यहाँ पे प्रोवाइड की है, that is suppose a rank list of targets, ठीक है, rank list of proteins. Now we can say कि other than these are the proteins on which this drug can act, ठीक है. Now these recommendations of course they have to be followed by the wet lab experiments in which you can detect you can predict for the side effects too, because these separate targets जो multiple targets आपने यहाँ पर recommend किए हैं, these targets can be responsible for the side effects. Okay, maybe that is a side effect, maybe it is not a side effect, so that may initiate, ठीक है, वो यहाँ पर inhibit कर सकते हैं, they may result into some side effects. So you have to मतलब you can use these results in predicting the side effects too. So this is the basis. Okay, you can say in the hierarchy, if you say at the root stands the drug target interaction prediction problem, or you can say the drug protein or the compound protein interaction prediction problem. Above it, okay, these are the problems. If we talk about the challenges of implementing an RS in medicine and biology, these are the basic challenges that have to be taken care of. You can see the lack of standard formats. Basically, when we speak about this challenge, ये basically क्यों होता है? Because standard formats, if we talk about such kind of format, we don't have the EMR records that have been implemented in India. We don't have a common format. So whatever records you are storing, they are very unstructured format. So it's very difficult to calculate the similarities and all. So you have to have a standard format जिसके basis पे हम यहाँ पर similarity निकाल सकते हैं. So for deploying a recommendation system, you need to have a standard format. Okay, and open source data in medicine, if we talk about the patient data, that is not open source. It's not available. ठीक है, तो यहाँ पर it's not available for free. You can't utilize for further analysis. ठीक है, आप research में भी अगर काम में लेते हैं तो that is not an open source data. ठीक है, so you have to take care about these challenges. ठीक है, next is the privacy. No patient would like to share their data, ठीक है, in the open source format. तो यहाँ पे privacy is a concern. Basically, probably in medicine, in medicine में हम जब भी यहाँ पे recommendation system deploy करेंगे, of course, you want a great amount of data. On that basis only you can generate fruitful recommendations that are relevant also. But in that case, you need to have data. ठीक है, तो privacy would be a concern. if we talk about the medicine area. Explainability and transparency, जब भी हम मेडिसिन की बात करते हैं, whatever recommendations have been suggested, जो भी हमने बताए हैं, तो you should be able to explain those recommendations. Suppose जैसे आप डीप लर्निंग यहाँ पे काम में ले रहे हैं, but you have used some images, ठीक है, आपने यहाँ पर निकाल दिए results, may be accurate, may be reliable, but you will not be able to explain what kind of things happen, है ना, हम ये क्यों कर रहे हैं, है ना. तो कोलैबोरेटिव फिल्टरिंग, सपोज यू हैव कोलैबोरेटिव फिल्टरिंग, जिसमें यहाँ पे यू कैन गिव द रीजनिंग, कंटेंट बेस पे भी हम रीजनिंग दे सकते हैं, बिकॉज दीज आर कंटेंट्स, है ना, दीज आर द फीचर्स दैट वर मैच्ड विद योर प्रोफाइल, है ना. तो ये यहाँ पर एक्सप्लेनेबल होना चाहिए. वट एवर सिस्टम या वट एवर एल्गोरिदम यू आर टेकिंग अप, you should be able to explain your recommendations. ठीक है, so this is a concern that should be taken care of. Next is the dependency on the contextual factors. You have to be concerned with these environmental factors. ठीक है, lifestyle, ठीक है, environment अगर हम कहें, climate कहें, weather कहें, ठीक है, gender कहें, तो these are the contextual factors that have to be taken care of while implementing an RS in medicine area. अगर यहाँ पे हम बात करते हैं तो बायोलॉजी की या ड्रग टारगेट इंट्रैक्शन प्रिडिक्शन प्रॉब्लम ही अगर हम लेते हैं, तो यू हैव इनअवेलेबिलिटी आपके पास है, दैट इज एक्सपेरिमेंटली वेरीफाइड एंड वैलिडेटेड डेटा दैट इज अवेलेबल, बट यू डोंट हैव, मतलब वहाँ पे एक्सपेरिमेंटेशंस हैं, ठीक है, आपके ओपन सोर्स डेटाबेसेस में मिल जाएंगे, बट यू डोंट हैव द नेगेटिव सैम्पल्स. जैसे हम अगर क्लासिफिकेशन की बात करें, because this problem can be taken up as a classification or it can be taken up as a regression based problem also. तो दोनों में से जो भी यहाँ पर हम बात करेंगे, तो क्लासिफिकेशन बेस्ड की बात करते हैं, तो हम यहाँ पे बात कर रहे हैं कि whether there will be an interaction or not, ठीक है, और उसके बेसिस पे हम रिकमेंडेशंस दे रहे हैं. अगर हम रिग्रेशन बेस्ड की बात कर रहे हैं, तो हम वी आर टॉकिंग अबाउट द बाइंडिंग एफिनिटी बिटवीन द ड्रग्स एंड द प्रोटीन्स. सो हाई अदर, मतलब जहाँ पे बाइंडिंग एफिनिटी आपके पास स्कोर्स होंगी, ठीक है, तो ऐसे डेटाबेसेस भी हैं हमारे पास जहाँ से हम बाइंडिंग एफिनिटी स्कोर्स भी निकाल सकते हैं. तो दीज स्कोर्स स्पेसिफाई द वेटेड थिंग, जो आप यहाँ पे एजेस की बात कर रहे थे इन द प्रीवियस स्लाइड्स, ठीक है, दीज एजेस आर वेटेड, ठीक है, दे आर वेटेड बाय द एफिनिटी स्कोर्स बिटवीन द ड्रग्स एंड द टारगेट्स, हाउ मच दैट प्रोटीन वुड बाइंड टू दैट ड्रग, है ना, ठीक है, तो वो यहाँ पर बाइंडिंग एफिनिटी से आपको मिल जाएगा. ठीक है, अब अगर हम बात करेंगे तो दीज आर द बेसिक चैलेंजेस जो आपको यहाँ पर यू हैव टू नीड टू address while implementing an RS within this domain. If we talk about the DTI problem, okay, because this is my problem, so I'll be taking up an example for this, in which we are taking up the drugs. Okay, we are extracting the drug features, we are extracting the target features, and we are preparing a drug target interaction data. ठीक है, that interaction data set will be prepared. After that we'll load it, ठीक है, we'll build a classification model both using Jupyter and Orange. ठीक है, तो Orange is an interesting tool. Maybe some of you must have not used this tool earlier, but it's a very interesting tool. जो यहाँ पे अगर हम बायोइन्फॉर्मेटिक्स की बात करते हैं या कंप्यूटेशनल बायोलॉजी की बात करते हैं, फॉर दोज पर्सन्स इट्स वेरी यूजफुल, because at the initial level, even if I want to use some machine learning model, या मैं यहाँ पर सिर्फ I only want to just see कि whether the accuracy will be as per my parameter or not, तो हम यहाँ पर सिर्फ initial level पे we can go for this kind of tool. Okay, so that gives you a basic workflow that can be designed. So next I'll just share my screen. You can. Is it visible to all?
Yes, Ashita. Yeah, thank you. Yeah.
Okay, this is the data set that I have downloaded. You can search it on through the research papers. Basically, it was given by Davis et al. Davis data set के नाम से यहाँ पर हम search कर सकते हैं. There are two kind of data sets. Basically, this is a regression based data set in which you can see here the first column. The first column are the drugs. ठीक है, जो drug की यहाँ पर PubChem IDs हैं basically. These are 68 drugs, total 68 drugs that have been listed out there. So this data set was initially provided by the researcher only. So I have only used this data set, and these are the targets, these are the gene IDs, these are the gene names that have been extracted from the UniProt database, and these are the affinity scores. These are the Dissociation constant, that is the KD values. These are the binding affinity scores that have been listed in each and every. So this is a kind of pivot table आप कह सकते हैं यहाँ पर. तो drug 1 and the target 1, drug 1 target 2, drug 1 target 3 and so on. So जितने भी यहाँ पे drugs and targets listed हैं for each and every one, ठीक है, आपके पास M अगर 68 drugs, 68 cross 442 proteins, these have been taken. So total 30,056 binding affinity scores are there in this matrix. Okay, so हर drug and हर target के बीच में there is an interaction that has been verified, that has been experimentally verified, of course, है ना. तो यहाँ पे 10,000, 320 जो भी आप देख रहे हैं, these are the dissociation constants that have been verified and validated by this researcher. So they had employed this data set for the drug target interaction prediction problem. So now what we have to do is, I can't show you the code for this purpose, but you can do it from PubChem, because they have an API in which you can extract these smiles for each PubChem ID. You can extract these smiles, okay, from just by querying the database. But आपको वहाँ पे PubChem, मतलब वहाँ पे you don't have to search it manually, है ना. तो ये आप कोड से निकाल सकते हो. तो these rows have been extracted for me. Each and every drug I have extracted the particular smiles, and the targets for each protein I have extracted the particular protein sequence that has been extracted from the unit prod ID. ठीक है, तो ये आप यहाँ से एक्सट्रैक्ट कर सकते हैं. दिस कैन बी इजीली डन प्रोग्रामेटिकली इन पाइथन. So ठीक है, I'll not be showing you this code, but you can do it. It's a small line of 3 to 4 lines of code, and you can extract in the form of CSV, and these sequences and these smiles would be used further in plotting the model, and making that machine learning model. And these are the affinity scores. ठीक है, these are 30,056 cells. ठीक है, जो यहाँ पर affinity आपको दिख रही है.
Harshta ma'am, your present screen is not visible.
Okay, just a minute. Is it visible now?
I don't know why it is again reflect your picture. Once it shows the Excel
No.
and then it reflects it.
I think it was visible,
Okay. Alright, so basically it's showing you are presenting to everyone. Okay. Is it visible now? Okay.
visible. Please go ahead. Yeah.
Okay. Okay, sir. In the next, this is the data set that I have employed. This is the latest data set. You can search it in the papers. There are 3 sheets here. We have some certain drugs, 68 number of drugs, 442 proteins, and they have been extracted from the UniProt database. Drugs have been extracted from the PubChem, and the affinity scores are provided. So this is the regression based data set that we are employing for building a classification model. ये हमने इसलिए किया है because classification में यहाँ पर हम basically what we do is, in the classification based data sets we have only 1 and 0 values here. Okay, 1 and values, 1 and 0, we don't know exactly what are zeros. Okay, zeros maybe they are non-interactions, maybe they are not verified, that's why they are termed as non-interactions. So what I have done is, I've employed a regression-based data set. Okay, we'll be utilizing this data set while implementing a classification model. So in the next thing, this is the Excel that I have. The first thing that was to extract the drug features. So I have used the ChemDes server. You can search this online. ChemDes server is available online, in which you have an aggregation. मतलब वहाँ पे बहुत सारे आपके पास separate tools हैं. You have RDKit there, paddle descriptors there. ठीक है, तो you can search for a particular one. So I have used the paddle CDK descriptors. These are the descriptors that have been यहाँ पे हमने extract किए हैं. This is done simply using the smiles file. तो यहाँ पे जितने भी smile IDs थे, I have formed a CSV there. I have uploaded that file, and after that I get this Excel in the form of this Excel in which we have the CDK descriptors. Okay, these are the 275 descriptors that I've used, but this is only one category. There are several more categories that you can use. Of course, say Jayaraman sir said you can go for the feature selection approach also. I haven't used the feature selection as per in this example, but you can go for that purpose also, because feature selection has to be done if you are using such a great number of features. So which feature is basically contributing to the model building, or which features are correlated, they need to be screened initially. Okay, so this is the basic process that needs to be done on a prior basis. That is a part of pre-processing. So I have downloaded this Excel using the SMILES ID of each and every drug. So these 68 drugs, for each of these drugs, I have these features or the descriptors that I have downloaded it as a file, and I have used an online tool for this. You can also use the RDKit packages also that are available there in our. Okay, RCPI package is also available that can be used for calculating the descriptors, drug descriptors and the protein descriptors both at the same time. So I have used differently. यहाँ पे हमने पैडल डिस्क्रिप्टर काम में लिए हैं. For the protein descriptors, I have used the PyPro package. ठीक है, for that purpose I'll show you one file. I hope everybody can see the file.
Yes, it is visible.
This is the ProPy is the package. This is the package that is available for Python. It is a Python based package. You need to install it from pip. ठीक है, आप pip से यहाँ पे install कर सकते हैं ProPy. And after installing this package, you can use its classes there. तो यहाँ पे relevant classes हैं and relevant functions are there for extracting the particular features that you want for the proteins. So for the proteins, I have used two descriptors, two classes of the descriptors. That is, one is the CTD, that is composition, transition and decomposition descriptors. And the second one is the dipeptide composition descriptors जो हमने use किए हैं और ये हमने extract किए हैं. तो this is only a relevant example. Of course, इसमें जो भी features आप category यहाँ पे काम में ले रहे हो, for that purpose you need to have feature processing. But I have used only two kind of features, that is CTD, one category, and the second category is the dipeptide composition category. Okay, for extracting this thing from this package, आपको यहाँ पर what you have to do is you have to supply the raw sequences, protein sequences there. You have to open this file, you have to read the CSV file. Next, row by row, we'll take one sequence at a time and we'll form a data frame out of it. And after making a data frame, हम लोग क्या करना है? We have to extract all the descriptors, CTD descriptors for each and every row. So these will be appended to this data frame. ठीक है, once, and after that हम यहाँ पे second category पे जाएंगे. We'll be extracting the dipeptide composition descriptors also. Okay, इसमें यहाँ पर basically what it does is these are the built-in functions that create a dictionary object. From that dictionary object, I have created a data frame, and after that we have two data frames, that is appended data 1 and appended data 2. These are the two data frames that have been created out of it. Now these two data frames are the two categories of descriptors have been generated. Now what you have to do is you have to concatenate these two kind of descriptors. Okay, because suppose जैसे यहाँ पे 100 है, यहाँ पे 250 है, तो these have to be joined together. So for concatenating these two, again यहाँ पर हम क्या कर सकते हैं? We can do two things. ठीक है, this is the commented one, but you can go for this code also. यहाँ पे दो टेक्निक है. One is through the use of pandas. आप यहाँ पे data frames को ही concatenate कर सकते हैं. The second one is
Harshita, excuse me. I apologize, Harshita. I'm sorry to disturb you. You know, we also have got some non-native English speakers, you know, from US and
Okay.
South America attending as well. So would you mind speaking in English? Yeah, please.
Okay.
I apologize. Thank you.
Okay. No, never mind. You can also go for the NumPy array in which we can concatenate two arrays. So for this purpose, either you can use these data frames. You have to convert it to two separate arrays, and these two separate arrays can be again concatenated together. So one form of joining these two arrays is like you can join two arrays. If you know, you can do this like while iterating through an array, or you can append two data frames together, because this is one of the most this is a better approach. You can go for concatenating these two arrays together, that is appended data 1 and appended data 2. These were the two data frames that we had created, and now we have joined it to, into a single data frame, that is a data frame in which we have the protein descriptors both from individually from both the categories, that is dipeptide composition descriptors as well as the CTD descriptors. So these again form a single data frame. Now you can save it to a CSV file if you want. So this can be done. So here I have also, you can save it separately if you want. But otherwise, what you can do is you can read the drug descriptors. Now what you have to do is protein descriptors have been extracted. Now what we'll do is we'll form the drug protein pairs. Now why this is needed? Basically, what we have is we have the affinity scores. Each drug is binding with a target with a specific score. Okay, so that score has to be taken up. Now we are forming a data set in which we have drug features, we have protein features, and they have been paired together. So each drug has been paired with each and every target, each and every other target. So you can see 68 drugs cross or 442 proteins. So they have to be concatenated feature wise also. So what we'll do is, first of all, we have to clean our data. So transformation, what we are doing is, we can see in the drug descriptor data, we have some NAN values also, some blank cells also, and some cells which are having some columns which are having only zeros. And some columns are there in which we have only unique value. You have the same values in the single column. So those columns can be dropped. We can drop those columns, we can remove those columns either. So I have done these steps for removing the NAN values. Plus we have replaced the zero values therein, and we have also we have also dropped the columns where the unique values are less than equal to 2. So this has been done as a part of pre-processing for the drug descriptor data, file. Next, we have to remove the NaN values also, that we have also removed. Now the next thing is we want to form the drug protein pairs. So these need to be formed using the combine operation. So we'll be forming how many number of combinations will be formed? That is 68 cross 442 numbers. That is 30,056 total rows would be created, because you are pairing up each drug with each and every other protein. So this code will iterate with every protein in this data frame. Again, this will iterate in drug in every drug data, and after that we'll be concatenating these rows together, that is the protein and the drugs together, and will form a particular Excel. So we have after that, when we concatenate, we combine these all things. Okay, after we have formed all the drug protein pairs, okay, we'll save this to a CSV file. So we have a CSV file in which we have the drug protein features formed. Okay, so we have all the independent variables there. Okay, in this file. So we have formed the drug protein pairs. Now this is the thing that we have created. But for a classification model, supervised learning model, we also want a target variable. For that target variable, what we have done is we have read the affinity file. Now this affinity file only contains the affinity scores of the drugs and the proteins. So what I have done is we have converted, this was a matrix kind of thing, we have flattened it to a single array, one column like thing, and then after that we have formed a data frame in which what we have is we have a column that is known as affinity, AFF. That column signifies this is the affinity score that has been provided for each and every drug target pair, drug protein pair. So this affinity will be combined with that data frame. So we'll be reading this CSV file in which we had formed the drug protein pairs, and after that we'll be concatenating the drug protein pair with the drug affinity score. Now this DF affinity, this data frame, this is a single column data frame. This single column has been concatenated with this drug protein pair. Okay, so this has been concatenated. Now we have a complete data set in which we have the drug protein pairs plus we have the target variable. Now target variable is basically a score. Okay, it can be 10,000, it can be anything. It is a real valued score जो यहाँ पे that is provided there in the last column. Now what can we do is before applying, if I this is a decision tree that I have built out of it. But before that, what we have done is, since we are applying a classification model here, we are replacing all the affinity scores, all the values whose value is less than 10,000, they can be treated as 1, and all the values that are greater than equal to 10,000, they can be treated as 0. This has been done in most of the research papers. They have treated it as like all the affinity scores that are greater than less than 10,000, they have to be treated as 1. They indicate an interaction between the drug and the target. And if it is greater than equal to 10,000, there's a lesser probability that there will be an interaction. So they can be treated as 0. Now this 0 is something like it conveys something because it is based on some score. If you directly put a 0 there, that is you can't rely on a classification based data set. But this, since it's a regression based data set that has been provided by this researcher, so यहाँ what we can do is we can replace this value wherever it is greater than 10,000, we can replace this affinity score with a 0 there. So we'll have such kind of if we run it, we have these columns here, and the last column indicates the target variable, that is the affinity score. Okay, these are the only 4 rows that have been displayed here, 5 rows. But otherwise, these are the columns. These are the drug features followed by the protein features, and the last column indicates the target variable. So this will be predicted by the classification model, particular classification model that I use. The first one is the decision tree classifier that I built. Initially, we have scaled these values. We have done some pre-processing, and after that, we have just done a split here in the train test split, and we have generated. We have these are the accuracy scores with each and every parameter that we have tested with. So this is the decision tree classifier that has been built with a simple train test split. But of course, you can go for a cross validation score, cross validation method also if you are evaluating a machine learning model. In this, suppose like this is an SVM model that we have used. The same thing has been done here also, and we have made an SVM classifier here in which we have tested it. The accuracy is 62.80, but this can be of course increased by particular feature selection methods if we do appropriately beforehand. But this can be of course increased to a much greater extent. But of course, we can also go for the stratified k-fold cross validation, because stratified k-fold means here we are also handling the imbalance, class imbalance that happens with these imbalanced data sets, because drug target interaction data sets mostly are imbalanced. You have a greater number of 0 entries than the 1 entries. So there are lesser number of interactions than the non-interactions. Okay. So what happens is this will be affecting your classification accuracy. So you can go for the stratified k-fold cross validation also. The same thing can be done in the orange tool also. So if you take these things, you don't need to write all this code. Of course, you need to prepare your data set in advance, in which you have to have this affinity file ready with you, in which you have the target variable and you have the drug protein features concatenated in the concatenated form, plus you have the target variable also. After that thing, all this Python code, all these machine learning models can also be run using the orange tool. Though you don't have very much hold over all the parameters there, But initially, if you run using the orange tool, you can get an appropriate results, and thereon you can test it in the Python also. You can change the parameters. Suppose you are working with SVM, you can change your kernels, what kernels you are employing. So that can be done easily. So that can be experimented with. But in Orange, you have a visualization workflow that can be created easily. That will be demonstrated by Dr. Amita Sharma, ma'am. Over to you, ma'am.
Thank you, Harshita, ma'am. Okay, I'll just share my screen. Harshita, can you just stop sharing
Sure.
the screen? I hope my screen is visible. Is it visible?
Yes, ma'am. Yes,
Okay.
ma'am.
So when you're going to install the Orange and when you with using the shortcut, you're going to open the Orange. The interface would be like something like this, in which you're going to find some menus, where menu will be file, edit, view, widget, options, and help. The rest important widgets are data, visual, model, evaluate, and rest which you can see in my screen. I have additionally added in my widget. For that, what you have to do, you have to go on the options, add-ons. Since it is showing some insufficient space, if you have sufficient space, you can easily add more widgets to your Orange tool. Okay. So firstly, before starting with the Orange interface, I'll just brief you why we can use the Orange tool. Firstly, Orange is open source tool, so you can easily use it at the beginning of your research work. Since if you are not very much friendly with Python or you're having some coding issues, first basic understanding of your results as well as for basic understanding of the various machine learning algorithms, you can use Orange tool just to start with. Also, sometimes in case of Python, if you don't have very good hardware and you get issues in executing your particular Python program with respect to huge amount of data, that case also Orange gives faster result in comparison to Python code. So that is the basic reason I am demonstrating this tool to you. Okay. So how can you start using the Orange tool? Definitely you have to download it. Whether you are working in Windows or Linux, you can download the Orange tool. You just simply type Orange on the Google and it will take you to the particular website, Orange tool website, and there you can download this tool. When you're going to download it, it has very simple step of installation. You can easily install it. After installation, when you'll open, you will get this kind of interface, which presently it is visible to you. Okay. So in files, we have general options. In edit, we have again cut, copy, paste options. View, we have different kind of zoom in and zoom out option. Then we have widgets, which you can see over here. These are all widgets. Then we have options. By using these options, you can simply add and remove the widgets available for your particular Orange platform. And then help, we have various kinds of help. You have video tutorials as well as you have already built-in workflows in this Orange, which you can easily adapt and work for your particular case study. So what Orange do for your case study? Orange generally generates a visual pipeline so that you can follow that pipeline and generate your result. See, in case of Python, what you do, you write step of codes like for importing your particular data, you'll generate a data frame, and then with that data frame, you're going to apply some pre-processing, and then after applying the pre-processing, you're going to split the data into test and train set, and then you're going to write the code for your modeling algorithm, but whether it is classification modeling or prediction modeling, whatever you are choosing, you're going to start with that particular modeling algorithm. Then you'll get the result. And for that results also, if you want to visualize that result, again you have to write the code for the visualization part. But in case of orange, you can do it in very easy way. I'll just show you how you're going to start. First, I'll explain you all what are these widgets are. First, we have data widget. When you click on data widget, you'll find number of options available over here. First, options are basically for importing the various kind of data. If you have a simple file, you can use this option. If you have CSV file, you can use this option. If you have special kind of data set, then you can use it. Then if you want to use SQL table, then you can use SQL table option. After that, the remaining options are basically to make a subset from the given data which you have uploaded in your orange. If you want to just get the information of data, you have data info. If you want to find a smaller sample using sampler technique, then you can use this option. And similarly, we have many other options which you can use as per your requirement. First step to start with is to select a particular file. You have to simply drag and drop on this particular dashboard, which is basically a panel. This white screen is a panel where you are going to create a pipeline for your case study. So firstly, in the case study, you are going to import your file. For that, you will just drag the file option and put on your panel. Since I have not uploaded any data, for uploading, you have to double click it. It will automatically tell you which data is currently available. There are certain data sets which is already present in the library of Orange, which you can easily use in for just for beginning with the Orange. And later you can upload your own data. For uploading your data, you can use this option. Click on this, and from a particular folder, you can select your data set. Like in this case, I want to select the ARES data set, which is very popular in machine learning. What this interface shows, it shows what are the various columns available in the data set. As in this case of ARES, we have following columns. We have 5 columns, out of which first 4 are basically the features of the particular item, and the 5th is basically the target variable or the predictor which we want to predict, which is again a kind of categorical data set. So this is the basic idea what data is all about, and also it gives you the details about the number of instances present in your data. This is the simple way you can select the file, which is the first way. Okay. Now in second step, what you have to do, you can just, if you want to visualize your data or you want to find the scattered plot from the data, then there are other options available in visualization widget. Using this visualization widget, you can get the different kind of plotting. Before moving towards visualization, I'll just tell you about the data table also. Data table is very interesting option. With this, you can simply view your data in a tabular format. So this is the way you can see, again the information is same but it's shown in a tabular format. It also gives you the details of the missing data. If there is any missing data, it will show you here that you can recorrect them. Now, if you want to move for further visualization part, for example, you want to place a scattered plot. You have to again select the scattered plot, connect the file with your scattered plot. These lines are basically the connector which connect you from one option to another. When you click on this, it will simply show you the scattered plot diagram. At present, this scattered plot is being designed between the petal length and sepal length and sepal width. You can also change the x-axis and y-axis with respect to your choice. For example, I want to plot scattered plot between the petal length and sepal length. This is the scattered plot. Okay, and also you can change this shape if your data is heterogeneous and you want to depict the data in various colors or various sizes. You can use the various options with respect to your choice. Okay. And we also have box plot, which we generally use to identify the outliers in our data set. Here it will give the outlier with respect to each field. You can select the particular field and get the box plot, and from this you can read the box plot and identify your outliers. Okay. After this visualization, you have many other visualization also. But it depends upon your case study, what kind of case study you have taken, then you can select the particular visualization. In case of data widget, we also have the feature construction option where you can construct your own feature. And also we have feature statistics where you can just simply analyze your features. There is one more interesting option in this, which is basically paint data. Paint data option is very interesting. You can simply paint your data items. How? When you click on this, initially it will show two categories, C1 and C2. After selecting the particular category, you can plot the data randomly just to generate a random data for a random study. Okay. Similarly, you can select any other class. If you want to add more classes, you can use this plus option and get more classes and plot it according to your requirement. After doing this, you can simply close it, and again you can use the data table option to get your data. See, this is the data which I have painted, and now it is being shown in the tabular form. So this is another important feature which you can use simply to generate your data. I'll just remove this. Now let's focus in building a machine learning model. So since we have file, we also have scattered plot, box plot. I'll just remove it. We have already seen what our data look like in a tabular form. Now if I want to do some machine learning on our data, specifically the supervised learning, then we have a model widget. It contains number of machine learning algorithms. So again, what you have to do, you have to select a particular model, place it on the panel by simply dragging and dropping it. For example, I take two model. I'll just perform first in on this SVM model. I'll just connect my data with this SVM model. Now to get the result from this SVM model, this SVM also has certain parameters which you can set according to your requirement. For example, you want to change the kernel, you can change the kernel. And if you want to modify the iteration limit, you can modify the iteration limit, but it's very specific. If you set the wrong value, then result will won't be generated. So try to be the specific, or don't try to set any parameters, keep it like this only. Then we have, after adding the model, we going to work on result which it going to generate. For that, we are going to use the evaluate widget, which has number of options. The first is the test and score option, and second is the prediction option. So in this case, since I am using the complete data and passing to the model, I can simply test and get the score for my model. I'll place it like this. In this case, I'm going to connect it with the model as well as with the data. When you click on this particular test and score, it will show you the name of the model, evaluation result, which contains the name of the model, AUC value, accuracy value, then we have F1, then we have precision, then we have recall. Okay, so this is a simple way you can generate the result using the test and score. Now, if in case I want to work upon more than one model, for example, I want to work upon random forest also, I'm going to simply connect my file with the random forest and then random forest with my test score. Now again, we'll see the result. Now here you can simply compare your models using this table, and you will get to know which model is working better in your particular data. Okay, and also there are other options available. If you have, since I'm using the simple training data where we have placed the values as well as we have the values for features as well as for values of the target. So we are working on the trained data. I selected this. If you want to work on test data, you're going to select this option. Now, if I want to generate the confusion matrix, since this is a classification problem, for that you are going to use the confusion matrix. You will connect the test score with the confusion matrix, and then you can simply click on this. Now here you can see the confusion matrix for each of the model. Okay, in this case, SVM model, the number of instance, based on the number of instance, this confusion matrix is generated. Here you can see the number of data set available and how they are classified. Okay, it also show which is basically misclassified. If you just bring your cursor on particular field, if you want to change in form of proportion, you can use this option. It will show you in the percentage form. Similarly, if you want to see in case of random forest and click it, it will display the random forest confusion matrix. Okay. Now, if you want to generate the ROC, you can simply click it and connect it with your test score. It will generate the ROC curve, and for different target, you can simply select the different ROC curve. Okay. If you want to download this or want to save this, there is a small option over here which you can use to save your file or particular image. Click on this and then you can save on the desired folder where you want to record your results. Okay, the result will be saved in PNG by default. You can also select it in a PDF format also. Okay, this is a small demonstration how you can work on Orange. If you want to work on machine learning algorithm, it can compare more than models, more than one model. You can easily drag and drop any of the model in any number of model, and then you can work upon it. Now, in case if you have some test data also, and since this is being worked on the training data, and you want to save this model, okay, so for that you have to do, for example, I want to save the random forest model. Since I have worked on test data, on train data, now I want to use it for test data. What I'm going to do, I'll just simply select it and drag it. It will show me some of the options, or it's better we can use this save model option and bring it over here and connect it. After connecting it, I'll just click on this, double click on this and save the model by giving some particular name. For example, I want to save it on my desktop. Workshop. Okay. I just want to give name to this model 2 random forest. Okay. This is the name which I have given to my model. Save. Okay. Now this is the way you can save the model. Now for if you have some test data, for example, if you have some test data and you have to apply the same model on the test data, how are you going to apply? I'll just demonstrate you in this panel only. I'll just again take a file option. This time also I'm using the same arrays data only. I'm not generating any other data set. Okay. And then what I have to do, I have to use this model which I have saved. So for that, I'm going to use the load model option. I'll click this option, double click it, and I'll go to browse the option. Desktop, yes. workshop. This is the model which I have created. I'll click on this, open it, okay, and then reload it and then close it. The model is being uploaded. Now in this case, what I'm going to do, I'm going to use the prediction option rather than the test score option. Okay, I'll connect my file with prediction as well as I'm going to collect my model with this prediction. Now what are you going to do? When you will click on this, you will find the value generated by the particular data set. So the actual value and the generated value you can easily see. This is the actual value which is present in my data set, and random forest has generated this value. So what has done? It has given the value to each category 0, 1 or 0, 1, 2 or something like that, and according to that value, it has generated the value for our data set. So you can easily compare your value, and also you can see the accuracy and the precision it has generated in the case of test data. So this is a simple way you can generate the model for your test data also. Okay, this is all about machine learning part. I'll just give you some more details about the orange. There are certain other widgets available in the orange. We have Bioinformatics widget, which contains several options. Since these options are a little complicated, I'm not covering it over here, but I'm just giving you idea what kind of options it has. The initial options, we have various data sets where you can simply develop the data from the data set. And for example, in case of geodata, I'll just drag this over here. When you click on this geodata set, you'll find various number of data sets available, and that data set you can use for your study. For example, I take some GDS1112 data set. I'll select this, okay, and close it. And I just want to know the information about this particular data set. At present it is uploading. I'll take genes. This is gene information, and connect both of them. Now here you can see what the values this particular data set contains. So this is the way you can also utilize the data set available in the available bioinformatic data set, and you can use it for your further analysis. So in the presentation which Harshita shared, she demonstrated the problem of drug target interaction. I'll just show you how we develop that particular problem in Orange. I'll just open my model. One more important thing, when you are designing this particular pipeline, you can simply save it using the file, save as option, and you can save that particular pipeline with your name. For example, Aris pipeline. Okay, for your future reference. Okay. Now, if I want to demonstrate you about the model, drug target interaction model. I will just open it. Okay. I'll just reload it. Are you able to see my screen? Is it visible?
Yes, very much, Amita ma'am. Thank you. Yeah.
Which model is visible? It is the file workshop underscore decimal models.
Yes, ma'am.
This file is okay.
It's visible.
Okay. So in this case, see, I have used the file option. In this file option, again, I'll just show you. I have uploaded it using the browse option. This is the file which Harshita used for it, the analysis she demonstrated in the Python. The same file I have used. You can see the number of instances over here, number of features, and there are no missing values in this. Okay. And also the important thing is with this option, you can set the target variable. Since in this example, I have already set, you can set any other variable also as a target variable if your problem allows it. You can simply go and select the particular variable as a target variable by selecting the feature option, that is basically the role of the variable in particular data set, by clicking this option, and you can select the particular role for particular variable. If you want to skip the particular variable, you can also again set the role according to the given choices. Okay. Now the second thing, again I have used the data table option just to demonstrate the data in my tabular format. Here you can again see the data in the tabular format. This is the target variable and other are the features of drug and protein. Now in this case, see, I have used four prediction models, machine learning models, and I have tried to compare those models using test score. Now here you can simply see the values. According to this, we can see random forest is giving the precise result, and in case of accuracy also it is much higher than other algorithms. So this is a simple way you can just see what kind of result being generated by the each model. Now I'll just tell you in this case study, when Harshita was doing this case study using Python, it was taking number of hours, but in case of Orange, we get the result in hardly 20 minutes. So at least we were able to understand what the result we are generating from our case study. Later we compared the result which we derived from Python as well as from this particular tool. Also, as I already mentioned, you can generate the confusion matrix also. See, by this you can see the confusion matrix of each model, and also ROC curve. Here you can see a beautiful ROC curve for each target. For one, it is like this. For zero, like this. Okay. I think this is all about Orange. If you have any other query related with Orange, you can ask me. I'll just stop sharing my screen now.
Ma'am, I want to ask a question.
Yes, ma'am.
Was there any difference in the results generated from Orange and Python individually?
See, in case of drug target, Harshita will tell. In case of other case study, as I worked upon Aedes and cancer data set, there was minute difference, not much which can be recorded. Harshita, will you tell about the
No, ma'am,
case study?
exactly the results were exactly the same. मतलब थोड़ा सा points में difference हो सकता है, but otherwise the accuracy was comparatively the same in both the tools. But in Orange, of course, you don't have to wait for a large data set. Also, you can provide that data set, but within minutes you will get the answer. So exactly you can see what kind of accuracy or what kind of recall precision these parameters are varying. And after that, you can go to Python and you can of course go for other parameters also. If you want to change this algorithm or you want to experiment with some other kind of things like pre-processing needs to be done, so that can also be done in Orange. But if you have to have a complete hold on your data sets and everything like that, for that purpose you have to go to the Python.
See, in case of Orange, what is there? You just initially we don't know which model is suitable for our
Yeah,
particular
exactly.
data set. For that uh, answer, you can go for Orange. It will easily lead you to the better solution that this particular model is suitable for your data. For that purpose, you can use Orange too. Okay.
Thank you, ma'am. I
Yes.
have a small question again, ma'am.
Yes, please.
Ma'am, what was the basis of this affinity score? Means, what are the specific value we need to focus on for pre-processing and generating those affinity scores?
Basically, Raju, basically, we have some affinity scores that have already been provided in the databases. Now, these are all validated scores. I'm not working on the wet lab experiments and all because they have been generated through these experiments. So you can't, you can have to take these values. You can build your model upon that. But specifically in the research, it has been said if the value, the binding affinity score has to be lesser. Lesser the value, the greater is the binding affinity. So it's said like this. So we can take the affinity scores, we can transform them into a classification based data set also, or taking up a threshold like thing. Here we have taken 10,000. So that is a threshold. If it is greater than equal to 10,000, then we think it's a non-interaction. And if it is less than 10,000, it can be an interaction. The lesser the value towards zero, that means good binding affinity is there. So the drug would be acting upon that protein, so you can have good results there. So that can be predicted if you are building a regression-based model. So you can have those results also there. But you have to have some feature selection out there because what features are contributing to that particular regression or binding affinity score while predicting or building a machine learning model, that has to be taken care of. So feature selection would be a great a point of research in which you have to select the prominent features of the drugs as well as of the proteins that will contribute to this model building. So you have to do it computationally. Of course, you can go after this, you can go for the wet lab experiments, but therein you'll have the fruitful results in case you have predicted them with a greater precision.
Okay, thank you, ma'am.
I think you'll agree with me that these are the excellent sessions that Amita and Harshita have really taken us forward. I think if you have any questions, please feel free to interact with them. They'll be there for the next 5-10 minutes.
I just want to add one more thing. In future, I'll definitely want to plan with Harshita definitely some small session on Orange, focusing on the bioinformatics vision, which we were not able to cover in this session.
Yeah. Yeah. Any questions, please, for Amita and Harshita, please. Yeah. Yes, Saud asked a question, can we have some scripts or data for that? Many scripts are usually available online. I'm not sure if this is a proprietary for both of them. Would you like to answer, Ramita or Rishita, on this?
Harshita will answer.
Yeah, data files, actually, these data sets are available online. But what I have done, the script that I have written for forming those drug target pairs, that may not be available. But of course, we can share it all lines of code. We can share it. But of course, you can find the data sets online. Davis dataset is a famous regression-based dataset for the compound protein prediction problem. Other than this, you can have the classification-based datasets also. It's provided by Yamanishi et al. And it's a very famous dataset. If you look upon the research papers, this is the famous dataset that has been used. Of course, and maybe in some other workshop, we can also tell you how to form your own
Yes,
data.
yes, that is very important.
That can be done from PubChem, Chembel. They provide you the APIs. You can directly query through the code and you can extract the data set relevant to you. Suppose if you want to have your COVID data set, so COVID related data is there in PubChem, Chembel. You can download it directly from there also, but you can do it through the Python also, through code also. So instead of querying there and having one record at a time, you can do it very comfortably through the code. So you'll have several parameters there, you can have filters there, and you can form your own data set. Because I have seen many research papers, they use their own data sets also. But they have been used in most of the researchers for validation purposes, we use such data sets. Any other question?
Yeah, I just have a small question, Harshita and Amita. So I know, taking, the regression analysis, from Orange, and again, checking, with the kind of a Python dictionary or Python libraries. How easy is it is, or how I wouldn't say easy, but how sensitive it is, to reach consensus, taking the Orange results and the maybe the Python results if we run in house, yeah, for any data set, yeah.
See, in this case, as we already know that we usually perform the data analysis using Veka. Again, we are using the already existing model, which is provided by the Veka, and we are using that for our data analysis. Similarly, in the case of Orange, the same thing applies. The only difference is it is providing you more visualization interface where you can visualize the thing. But the code is the same, which is being used by different tools. In case of
Right.
Python also, we are using the inbuilt library. We don't know what that library contains. We are just setting the parameters according to our choices. If you want to work purely in machine learning, then you have to design your own code for each model. That will be again difficult for everybody who is not having a computer science background or computer
Right.
engineering background. So in that case, I don't feel like in comparison it is every time okay whether you compare it with Reka or you compare it with Orange. They are giving the almost the same result and they are using the standard algorithms. So there are no issues in respect to the performance what they are giving.
Right, right. Yeah. Yeah, yeah. Thank you so much. Yeah. So, are there any questions for both of them, please? Yeah. Yeah. Sorry, Jairam sir. Yeah.
See, for the recommendation systems, they are using only activities as the this thing as a methodology. Or are there any other attributes which can be used? I'm asking. Yeah.
Pardon, sir, can you repeat the question, sir?
Question is, for recommendation systems in drug target systems, what are the qualities or the attributes which can be used for recommendations? Yeah. But other than what you have done. Yeah.
So actually, we would be working on the feature selection because what I think, what I have read till now is basically you should be able to select a set of features, particularly for the drugs and as well as for the proteins, okay, which features would be more suitable in predicting these values. So if even if you design a recommendation system on that purpose, for that purpose also, we need to have a selection criteria. What kind of features would contribute more and which features can be eliminated out of these set of features, because you have a range of descriptors that are provided for drugs as well as for proteins, because I have only taken a one subset out of it. But of course, we have to have a feature selection algorithm that you taught earlier in the first workshop. We may go for the genetic algorithm or such kind of things because they need to be done priorly as a pre-processing part. After that, we can of course build a model and we can have the predictions. We can have the recommendations, but they need to be improved, of course.
Okay. Yeah. We can discuss further later. I think. Yeah, I have more to discuss here.
Yes, sir, sure.
So, there are no questions. I think we should wholeheartedly thank Amita and Harshita for their wonderful time and energy. It's been an amazing talks, from both of them. Absolutely marvelous to hear both of them. Thank you so much for your time and energy, Amita and Harshita.
Thank you so much, sir.
Yeah, would you mind in all of you to switch on your video, please, for a quick photograph? Yeah. Yeah, yeah. These are all the machine learners, all the machine learner group team. Sundarda is also there from Singapore. Everyone, please One quick photograph with all smiling faces. I think I'm just, yeah. Yes. So thank you so much, Sage. Yes. Jayaraman sir is not visible. I am. Nidhi is here. How about yes, Sonal is also here. Yes, Shalini. Yes. Thank you so much. Thank you. So see you tomorrow. If someone is very fond of music, the music will play for the next 10-15 minutes. Okay. Thank you so much. Yeah. And please see you, tomorrow, for the workshop. Yeah.
Thank you so much.
Thank you, everyone.
Prashant, still there?
Yes, sir. 1 minute. I'm just. I wanted to entertain our live audience here.
Okay. You forgot to. The audience forgot to. Both of them are.
Yeah, please, sir.
Let me thank both of them for an excellent
Hello sir, how are you, Professor Jayaraman?
I'm fine.
Morning was good, but sometime I was able to follow you as I was in office full day.
Calm down. Your work. Yeah.
Yeah, I am working in office now. Yeah, it was